Hi, welcome to another episode of Chromatography Experts, where we discuss all things bioseparations. My name is Anston Lewis, Global Sales Manager at Tosoh Bioscience, Separation and Purification, and I'm joined by my colleagues Marissa and Abbie.
Hi, I'm Marissa Heino. I'm one of our commercial product managers covering our process solutions product lines. So these products include our Octave multi-column chromatography systems, our skill-packed pre-packed columns, and then also our process resins.
And I'm Abbie Breton. I'm the Manager of Application Development here at Tosoh Bioscience, as well as the Acting Application Scientist for our multi-column chromatography instruments.
Today, we're going to revisit the topic of MCC, but incorporate a bit more specific topic of viral clearance in bioprocessing. So, to kick us off, my first question to you guys will be, as we mentioned, we talked about MCC before, but Marissa, do you mind just briefly running through the principles of MCC just so the audience can be reacquainted with it?
Yeah, definitely. So I know I wasn't part of the first episode, so maybe I can bring a fresh perspective to what multi-column chromatography is. So I think the important thing to do is to first take a step back and kind of look at the bigger picture to really realize where MCC falls into bioprocessing, downstream bioprocessing, etc. So we hear a lot about process intensification, whether we're at conferences or talking to our customers. People are really interested in efficiency gains, cost savings, things like that. So when we're thinking about process intensification, there's different ways to intensify our processes, whether we're talking about intensifying in our upstream process, or what we're going to be focusing on today is a way to intensify your downstream process. So there's several different unit operations in the downstream process, and what we're going to be focusing on today is the chromatography unit operation. So in batch, we've traditionally been focused on the safety and product quality, and we've kind of got that down pat. And so now, like I said, people are really looking to intensify this unit operation. So multi-column chromatography is one way that we're able to intensify the chromatography step. And thinking about what multi-column chromatography is, I think Abbie did kind of run through what it is, but a really basic definition is that we have our separation steps occurring simultaneously across multiple columns.
Thanks for that, Marissa. Appreciate that. So can you speak a bit more to, I know, Abbie, we talked about this before in our last episode, but, I'm curious since we have a new guest, your perspective on, the specific benefits to our customers for MCC relative to batch.
Yeah, absolutely. So when thinking about MCC, there's a few different benefits that we do like to highlight. So the first one is going to be productivity gains. So when we're thinking about productivity, we think about productivity in terms of grams of product per liter of resin per hour of processing time. So we've actually seen up to a 300% increase in productivity compared to batch, which is really great. So when we have that increase in productivity, we also see a decrease in resin volume used. And so the typical application for MCC is going to be for your affinity capture step. So when we think about using less resin, we're actually going to be able to save a lot of money in terms of that expensive affinity resin that we're using. So that's been a real benefit to a lot of our customers. And then also kind of an added benefit to that is that we also see around a 40% decrease in buffer consumption as well for certain processes. And these numbers that I'm throwing out, they're not going to be exactly the same every time, but for a lot of the time, you are going to see significant increases in productivity, resin volume, and then also buffer usage as well.
Okay, so less time and less buffer and resin usage. Sounds like a win-win.
Yeah. So really efficient, and then also a lot of flexibility in terms of what you want to maximize. If you are looking to decrease that overall processing time, you have the option to do that. If you're really looking to decrease your resin volume, you have the option to do that as well. So MCC offers a lot of flexibility in terms of what you want to intensify in that chromatography step.
Okay, thank you, Marissa. So as you mentioned, Tosoh, we participate in the separation and purification of biomolecules. So today we're going to foray into a slightly different topic, viral clearance. And Abbie, I know you're well experienced in this space. So can you kind of speak to what does viral clearance mean? Like, why is that important in bioprocessing?
Yeah, so viral clearance is actually a requirement for any downstream processing of biologics, especially those that are being produced in mammalian cell expression systems that may actually contain endogenous retrovirus. So in downstream processing, and specifically viral clearance, there's two types of what we call steps, either dedicated or non-dedicated steps. So dedicated viral clearance steps are process steps that are added into our purification that are specifically designed to remove virus. So these are things like our low pH inactivations, solvent detergent inactivations, and then nanofiltration or viral filtration. And then we have steps that are called non-dedicated viral clearance steps. So these are our actual chromatography steps, which are in our process to remove other things like our host cell proteins, our DNA, aggregate, things like that. So these are, like I said, all of our chromatography steps, like our affinity, our ion exchangers. And these have varying levels of effectiveness to them. So we have anion exchange chromatography, which is typically very effective at removing viruses. And then we have our other chromatography steps, like our cation exchangers, our mixed mode HIC resins. Those are relatively effective. They're kind of condition dependent. And then we have our affinity resins, and those are really not effective at all at removing virus. But of course, it's a very important part of our purification process, and we do still usually test that for viral clearance. So that's just kind of like, just like a very generic overview of viral clearance.
Can you speak to maybe, if you can, like the challenges presented today by our customers when it comes to viral clearance specifically?
Yeah, so there's definitely, I would say, a lot of concerns from our customers moving from batch chromatography to multi-column chromatography and what that really means in this viral clearance space. One thing that I do want to point out is there are updated guidelines. They're the ICH Q13 guidelines, which are really giving guidance to the industry that are looking to move towards a continuous manufacturing of drug product and drug substance. And just to kind of really summarize it very quickly here, these recommendations, I would just say the recommendations that were previously outlined in the older version, the ICH Q5, are still applicable to continuous manufacturing. So there's no real additional guidelines here when you're switching to MCC. And I know that's been, like I said, a big concern for our customers. But one very specific concern that I've heard is I know we kind of mentioned this in the last podcast, that one of the ways that we also increase our productivity is by overloading our resin. Because we're able to load columns in series, we can overload that resin to about an 85% of its dynamic binding capacity. One of the concerns there, of course, is if we overload our resin, we could see some virus breakthrough in our product.
That'd be bad.
That would be really bad. Yeah, I mean, because if you have virus breakthrough in your product, your viral clearance numbers are lower when you go to file with the FDA. It doesn't look good for the FDA for your product. But we're not really worried about that here with the way we do MCC because we overload and then are loading in series. If there were to be any virus breakthrough on any of your columns, it's going to be caught by our subsequent columns and should not end up in either your elution or your flow through step there.
So thank you for that very, very detailed response. And compared to the last episode where we discussed MCC, I noticed for this specific topic, we're incorporating a lot more of the regulatory and quality type of concerns when it comes to bioprocessing. And for our younger or more inexperienced chromatographers or scientists out there, can you explain some of the terms that I heard you use, like ICH Q5? What is that, and why is that important?
Yeah, so ICH Q5, ICH stands for the International Council of Harmonization. So these are just really international guidelines that were put together by people in industry to really help guide our customers, people in this space that are producing drugs. So it doesn't just have viral clearance like guidelines, and that there's many different guidelines in these. So the previous version was called the Q5, and I think it was maybe 2 years ago, don't quote me on that, this new version, the Q13, came out, and it was specifically aimed at moving towards continuous manufacturing because we do see that is how industry is moving. And so there was definitely a need for these guidelines. But these are publicly available online. You can just, you can look them up. It's a relatively short document for it being, being a guideline. It's only about 40 pages long. So yeah.
You mentioned some concerns our customers might have from transitioning to MCC for viral clearance. Why would they want to? What is the bull case or the reason why they would transition over? What advantages would they have? using MCC as opposed to not?
I think I can probably start by just simply saying when we're transitioning to MCC, now we're introducing the use of more columns into the scenario. So that might just be, or introduce some hesitancy to our customers. Essentially, we're going from using one column to anywhere from 5 to 8 columns in a process. So that might cause some hesitancy there. And then on top of that, Abbie mentioned about how we are overloading our columns with feed. So that statement again might just cause some people to say, okay, well, how will that affect our process in terms of viral clearance? So I think that's kind of maybe a basic answer.
Okay.
To that.
And I would say, also just kind of playing off that, batch chromatography viral clearance study has been done with batch chromatography for years and years and years. And that is the way it's been done. So, this is I would say a big change. So that is definitely what the hesitancy is there.
And I think batch chromatography has been performed, for so long. Like Abbie said, I think even in the transition to MCC, people are hesitant. And when we're asking, now we're bringing in other factors like viral clearance. Again, we're just adding extra factors that people need to think about. So whatever we can do to provide them with the security that the transition to MCC is going to be as simple as possible and as safe and effective as their batch process is what we're hoping to do here.
Understood. Change is scary, right?
Yeah.
All types of change, and especially when it comes to heavily regulated industries like ours.
Yes.
So, I'm the sales manager here. I'm always concerned about how to make this a positive, right? Mm-hmm. How do I alleviate our customers' concerns? What about this transition using MCC for viral clearance? What advantages are there that should kind of quell the concerns that some may have to make this kind of change?
Yeah, so we can kind of point back to what Marissa was saying earlier, that some of the really big like efficiency gains here. So that is really going to be one of our big things, that there's these huge efficiency gains moving from batch to MCC chromatography. With kind of like the work that we're doing here with our viral clearance study that we will probably discuss here, we're really just showing that when you move there really is no difference between batch and MCC with your viral clearance. So it's not that there's, you're getting better or worse viral clearance there. You're getting the same results that you would see in batch, but with all those efficiency gains with your, time savings, resin savings, things like that.
I think that's a great segue. Abbie, let's get into this study. So Abbie, could you kind of lay out this really exciting experimental design? What were your thought process kind of, going into this, how you designed it? What factors did you have to incorporate to make sure you really got a really comprehensive study here?
Yeah. So with this study, the ultimate goal of the study really was to develop a 2-step purification platform to show a few different things. So satisfactory overall MAB recovery, our increased process productivity, like Marissa mentioned, but also effective viral clearance. Also, we really wanted to highlight the flexibility of these MCC systems. Like Marissa was saying earlier, our typical use case for these systems is that initial bind and elute affinity capture step. And, since that affinity resin is typically the most expensive resin, that's where we see the most productivity gains. That's why most of the examples we see are with that bind and elute capture step. But here I really wanted to show that we could use this system in not just a bind and elute mode, but also a flow-through mode. So I did choose an anion exchange resin for our polishing step. I chose our Toyo Pearl NH2 750F resin in flow-through mode to really show that the system was very flexible between bind and elute and flow-through mode. And I specifically chose that resin because one of the big problems that our industry sees when moving from our capture step to our polishing steps is in our elution fraction. We're usually seeing pretty high concentrations of salt due to those elution conditions. So because of that, most of the time we would need to dilute that product, which of course is going to lower your titer moving into your polishing steps. But this resin, the NH2 resin, was chosen specifically for that reason because it works really efficiently at a relatively low pH and also physiological salt conditions. So specifically for this purification, we ran at a pH of 6.5 and a 150 millimolar NaCl concentration, so physiological salt condition. So there was no need to dilute the dilute our material between the elution of our capture step and the polishing step. All we did there was adjusted the pH. Because the elution happens at a lower pH, we had to adjust that up to a pH of 6.5. We also did know from some previous work that was done a couple years ago that our NH2 resin is a relatively good resin for virus removal or for viral clearance. So we kind of knew going into that. Well, we figured going into this we were going to get some pretty good results. So that's really the reason for me choosing that resin. But then just go into like some actual details of the study. So for the protein A step, we used our high capacity protein A resin in our 5 mil pre-packed scale pack bio columns. So these are columns designed specifically for MCC systems. And we used 5 of those columns. So a total of 25 mils of resin there was used. To analyze at the end, we did we looked at aggregate content and monomer recovery in that. As well as host cell protein removal. So those things that Marissa had mentioned earlier, I had mentioned earlier. And then for the polishing NH2 resin, we used 3 1 mil skill pack bio columns. And like I said, in flow through mode with a relatively low pH and physiological salt conditions. And then to kind of touch on the viral clearance portion of this, here at Tosoh, we don't have labs that are capable of handling live virus. We're not, we don't really want to handle live virus here at all. So we really relied on mock virus kits from Cygnus for the actual spiking portion of this study. So we assessed the viral titer pre and post purification using those kits.
So that's very interesting. What isn't a mock virus kit? Is it a fake virus? Like, what is a mock virus kit?
Yeah, I mean, it pretty much is a fake virus. So they're really interesting kits. These are kits that contain, so they're non-infectious virus-like particles that mimic the properties of live infectious viruses. So there's 2 kits that are offered currently by Cygnus. There's a mock parvo kit and a mock retrovirus kit. And if you're unfamiliar with that for viral clearance, the parvovirus and retrovirus are 2 of the viruses that you need to show for at least your initial IND filing. You need to show clearance of. So these kits are really great because with viral clearance, most of the time you need live virus. Well, a lot of our customers don't have the capability of using live virus in their facilities, nor do they want live virus brought into their facilities, especially if they're manufacturing in those facilities. There's just too much risk involved there. Also, viral clearance studies can be prohibitively expensive and also like really time consuming. So these kits are super good when you're doing your initial, I would say, process development to really just get an idea of what your viral clearance numbers look like. Because if you think about, you develop this process, you pay a ton of money to have a viral clearance study done at a CRO, and you show like no clearance, and you have to go back to the drawing board and really like change. You have to optimize your process. Whereas you could do that all in your labs before you would go and pay for this viral clearance study. The viral clearance portion of the study was performed just like we would perform one at a CRO, just with these mock viruses. So we spiked in a known quantity of that mock virus into our starting material, took it through the purification process, and then tested the titer prior to and post purification. And I don't know if I mentioned earlier, this is these titers are determined via an immunoassay followed by RT-qPCR. So how that works is we have a microplate that's coated with an antibody to that mock parvovirus, and so we will put our samples into that microplate, and if there is the MVP, the mock virus present in that sample, it will bind to that plate. And then in further steps, there's a DNA conjugated anti-mock virus antibody that is added, and that will bind to those samples again that contain that mock virus. And then finally, there's qPCR that's done using TACMAN primers that uses a probe specific to that conjugated DNA. So that'll give us our final titers. So it's actually a relatively easy way to, and within a day you can get your answers there. As most of our clearance studies can take a couple days to get your answers there.
Yeah. Awesome. Before we move on, Abbie, I just wanted to kind of take it back. I think one question. So in talking about the setup of the two different processes. So for our capture protein A step, you talked about using 5 mil skill pack columns for that. And then for our polishing step, using the NH2 resin, we use 3 1 mil columns. So I was wondering if you could talk a little bit about how we're determining how many columns to use, what size columns, and then also maybe just talk a little bit about what our skill pack columns are.
Yeah, for sure. So I landed on 5 mil columns for the protein A, really based on the amount of like load material that I had to process there. I did have quite a large amount of load material. So I used those 5 mil columns, but I have used 1 mil columns in the past. But I would say there's a tool that we use, a software tool called our Method Wizard that I don't remember if I touched on this in the last podcast, but we can really use that. We can input things like our dynamic binding capacity of our resin, what we want to load our resin levels to, so that 85 to 90%. And that'll really give us like an idea of how many columns we should use to really maximize that efficiency. So that's kind of how we landed on those 5 columns for that. And I will say I've really, through most of my work, I've really optimized our protein A method to use a 5-column method. And with that, I know I touched on this in the last podcast, I will usually load 3 columns in series there. Yeah. And then the other 2 columns are, going for the subsequent steps at the same time. Now for the NH2, so there, if we think about we're going from a really large volume of feed for a protein A, and our elutions then are pretty small that come out of that. So that's why I went with the 1 mil columns there. I had much less material that I needed to process for my NH2. So there's really no reason to use 5 mil columns there. I use those 1 mil columns. And also we found that 3 columns, we were really able to maximize our efficiency with just using those 3 columns. So for that, I loaded 2 columns in series there. Then that final column was going through all of the subsequent steps, like the strip and sip and things like that. But then just to get to your second question about what are skill pack columns, realized, we didn't talk about that in the first podcast, but so Tosoh has a line of pre-packed columns for both batch and MCC. So they're called skill pack. So just regular skill pack are for our batch chromatography. And then we have what's called skill pack bio, and those are meant for our MCC applications. And especially for the MCC application columns, those are designed specifically for our systems. So they do have much shorter bed heights than our traditional batch chromatography columns, usually around 5 centimeters there.
Great question there, Marissa. Making sure we get to what are skill pack columns. So Abbie, thanks for giving us the whole outline of the experimental design. What were the results of the study and were they expected?
Yeah, so I can say just overall, yes, they were expected, but I'll dig into the results a little bit more. I am going to focus mainly on the NH2 polishing step here because that was really what we were trying to focus on with this study. And now let me just kind of explain a little bit more why. If you remember from earlier when I was talking about dedicated versus non-dedicated steps, I mentioned that affinity chromatography is really not effective at all at clearing virus from our steps. To be honest, when a lot of our customers are submitting viral clearance results to FDA, whoever your regulatory body is, for your IND, BLA filings, etc., the protein A step sometimes is not even included in that because you're not really even meeting that minimum threshold for to show viral clearance. So that being said, we really didn't include the protein A step in this initial viral clearance testing. But I will still talk about some of our quality attributes from that run. So as we mentioned, that we ran a 5-column purification method for this, and we ran our purification at a 30-second residence time, which is much faster than batch chromatography. There, we're typically running at a 4-minute residence time. We had a cycle time of 33 minutes. So, and we ran 3 cycles there. So in just under 2 hours, we were able to process over 3 grams of our monoclonal antibody with that 25 mils of resin. So that's a really big amount of antibody we were able to process in that little time. Our yield from here was 92%. And with the size exclusion chromatography results, we saw less than 5% aggregate. So really great results in this MCC run, very comparable to our what we would expect in a batch run. So regards to the NH2 step, We ran this at a 1 minute residence time, so a little slower than the Protein A, but still much faster than we would typically for batch chromatography. Again, around a 4 minute residence time. So our cycle time here was a bit longer, 52 minutes. We ran 5 cycles, so a few more than we did for the Protein A. But with that, we were able to process almost 2 grams of MAB in just over 4 hours using only 3 mils of resin. So that's a huge gain in productivity there compared to batch. To only use 3 mils of resin to process like 2 grams of MAB is really great. So our yield for the step was 94%, but most importantly here for our viral clearance, we showed complete clearance in this run. So our result here, we had a number of greater than or equal to 3.23 log reduction value. And that greater than or equal to symbol sign just means that we were under the limit of detection for this. So we're not able to give an exact number, but anything that's under the limit of detection is considered full clearance. We did do a batch chromatography run, like viral clearance run as well, just to make sure that it was comparable between. And we saw complete clearance there as well. Very comparable number. I believe it was 3.42 for that. So pretty much identical results between batch and MCC. So it really showed us that there's no difference for us between our batch chromatography and MCC when it comes to viral clearance. Especially getting back to that, really overloading those columns. We saw no virus breakthrough in those in that fraction that we tested.
Okay, so the clearance ability of batch with the efficiency gains
of MCC. Exactly. Yeah.
Wow.
Yeah.
Win-win situation once again. So thank you for going over that. So like the last episode, I'm always thinking forward, right? Based on these results, do you see implications? Because you mentioned I think customers right now, they outsource some of these viral clearance studies to contract organizations, things of that nature. Do you see implications to whether it's the business model currently of viral clearance studies and things of that nature, or just in general for bioprocessing with this type of results you got from the study that you can use MCC for viral clearance?
Yeah, I mean, from this, I definitely see that you could use MCC for viral clearance. So I don't think that there's a need to be concerned about, if you have an MCC instrument or if you want to move towards MCC, you could use this instrument for your viral clearance, for sure.
Marissa, you have anything to add here?
Yeah, so I think we've kind of talked about it, but I think the goal of this study was to show that we do have comparable results to batch in terms of viral clearance, those critical quality attributes, but with all the benefits of MCC. So when our customers and industry is transitioning to this new territory, whether it is just multi-column chromatography or we're maybe linking different unit operations, talking about continuous manufacturing, that MCC is only going to give you an advantage in this field. We're not worried about viral clearance or decreasing the safety of the drugs that we're manufacturing. I think the study is just showing us a lot of promise for people in industry feeling more confident in transitioning to MCC. And I think overall we're excited to continue to work with our customers to show them that this is not only an efficient and flexible new way of bioprocessing, but also safe.
That's a great dismount, Marissa. So to bring it back to something you mentioned earlier, continuous processing, can you kind of quickly just define what that is, and then where do we as Tosoh kind of participate or support in that space?
Yeah, absolutely. So again, like I was talking about in that bigger picture, when we're talking about process intensification, one of the ways to intensify the process is to actually have continuous manufacturing. And so that can look a little bit different from different people, different perspectives. So when we talk about continuous manufacturing, we're talking about integrating different unit operations in your manufacturing process. So that might look like a complete continuous process from your upstream, completely connected to your final product. That might be semi-continuous processing. Maybe you're only integrating some unit operations in the downstream process. But what it really looks like is linking different unit operations together to keep a process continuous. So Tosoh is interested in moving into the continuous manufacturing space, hoping to be one unit operation in that overall schematic. So we still are and are currently a single unit operation as the chromatography step, but we are looking to ensure that our Octave is able to integrate with other third-party systems in order to fit into that continuous schematic. So there are a few different ways that Tosoh is actively working towards this continuous goal. We do have a study called the eConti project, where we partnered with a few different Austrian companies, actually, in order to set up a fully continuous process. So we're not actually doing capture chromatography. In this study, we're actually manufacturing the protein A ligand from E. coli. So slightly different than our typical use case, but I still do think it shows the flexibility of what the Octave can do. So here we integrated 3 different unit operations in order to come out with that final product. And the Octave is, like I said before, being integrated with third-party unit operations in order to keep that continuous. So like I said, it's called the eConti project. There's a lot of information about it online, and we'd be happy to, share some information if anyone's interested.
Exciting stuff.
Yeah. And Tosoh, I think that's just like a little bit of a teaser about what Tosoh is going to have to offer in the next couple of months or so. So definitely stay tuned for that. We're excited for some new features to be coming out in our system.
All right. Thanks, Marissa. So thanks again, Abbie. It's always a pleasure. And Marissa, thanks for joining us today.
Thanks for having me.
Podcast. And that's going to wrap us up today for another episode of Chromatography Experts. I'm Anson Lewis. You guys have a great day.